Thank you very much. I thanks everybody to allow me to be here today. My presentation, I have to say, is the resume of all my previous research work that was done with the collaboration of many research institutions. I will mention just the main ones that are the Massachusetts Institute of Technology, the CNES, that is the French Spatial Agency, and the French National Research Institute, the CNRS. And of course, this work will function as a base and background for future projects and objectives of the Air Center. So this will be a very good example to show you how something that until a few years ago would never been imagined to be possible. How we can detect biodiversity just from space. We will be able to do it, and I will show you how. First, I want to introduce you the concept of biodiversity that we are using. Biodiversity is defined as the variety of diversity at all levels of the organization of life. And if we look at the ecosystem, we can identify genetic diversity, species, communities, and also assemblages of different ecosystems that forms the biomes. This diversity can be measured with different means, in particular molecular, morphological, or functional. And it's not automatic that all these measures will give the same results. Biodiversity is very, very important for the maintaining of all the ecosystem functions, to provide important ecosystem services to society, and to provide direct resource supplies. Unluckily, in these few decades, we are assisting more and more to a loss of biodiversity, not just on land, but also in the ocean, where we are not even already sure of how much we have discovered already about this biodiversity. In fact, our planet should be called more planet ocean than planet Earth, as 75% of the planet is composed by oceans, and the salt water composes 97% of this water. And the ocean is also responsible for 50% of the primary production, so it's more or less the same that is produced on land. The ocean is also responsible for the homeostasis of the planet, is responsible for climate variations and also changes, because it can act as a buffer for biogeochemical changes in the water, but also with the atmosphere. And it provides a lot of ecosystem services, as we said. Until a few decades ago, let's say, the ocean was considered to be a very homogeneous and not so interesting environment. Instead, with the possibility to observe it in a better way, so with more oceanographic campaigns and also the availability of special observations, we realized that the ocean is a very rich and dynamic environment, and moreover very heterogeneous. This heterogeneity is the thing that gives hard times to scientists, but also to politicians and economists afterwards, because it's not possible to easily sample it or know exactly what there is over there, because the extent is huge and it's very heterogeneous. So even diving procedures and campaigns would not give enough data. And even the best oceanographic campaigns, when they monitor it, they can sample water masses, as we can see here, that can be very different from each other. Here we have an example of a blooming, phytoplankton bloom, and we can see that if we sample in the lighter water mass, we will have a very different biological community, but also biochemical characteristics of the water masses from the ones that are in the darker side, where the blooming is not still present. Moreover, when we sample the water in the open ocean, we have a hard time to identify which species are there because most of the microbial species are unknown. Luckily, we had the improvement of some technologies since a few decades. Satellites are able to detect through the use of different light spectra different functional groups of organisms that are in the water. Basic satellite data that are used to describe the environment are now ocean color. As I showed you before, it allows to detect blooming of phytoplankton. As you can see in the first image on the left, is the region of the Patagonia with the confluence between the Brazilian and Malvinas current. Altimetry, that is used, in this case, it represents the same region to derive ocean fronts and oceanic currents, and the sea surface temperature, that is the basic characteristic to characterize biogeographic fronts. From this, we can have derivative products, so more elaborated products. In this case, I have an example that uses all the ocean color data, that is the FISAT algorithm. The FISAT algorithm is able to discriminate between different phytoplankton functional groups. In my study, I used the version who was able to discriminate until 6 groups, but now it was developed to discriminate until more than 100 functional groups. So the study is really going on and on. From altimetry, we can derivate Lyapunov exponents. Lyapunov exponents is a measure of the strength of the transport barriers, and this is a very good indicator of where the oceanic fronts forms and disappear and where they are stable and how much can be the passage of biological communities from one side of the front to the other for organisms that are not allowed to swim freely. And of course, we can also calculate temperature gradients and fronts. I used all this data, but in particular, the main driver I realized that was the ocean color, to try to understand how it would be possible to detect biodiversity hotspots in the whole ocean at the global level. Why I used this kind of tools? Because phytoplankton is very important because it's the base of the trophic web. And so if we can understand where are the hotspots of biodiversity and also biomass possibly for the base of the trophic web, we will have also an understanding of how will be the trophic interaction and so the functioning of the ecosystem in the different regions. As you can see, we have temporal variability because we are able to monitor the whole ocean for the whole year, and this is a great advantage of the satellite data. What we tried to do was to create an index based on this available data of diversity. Of course, it won't be one of the classical biodiversity indexes that are described as alpha, beta, and gamma diversity, so local, regional, and global diversity, but it will be more a mixture of the first two. In fact, we will detect regions that are more heterogeneous in terms of dominant phytoplankton functional groups and also of the whole characteristics of the water masses in terms of biogeochemical characteristics. And we will see what these change at the global level. So regions that appears with more color are considered more heterogeneous in terms of the environment and also biological community. The ones that are more homogeneous in colors will be the opposite. Of course, to validate this approach at the global level, it was not possible simply because there are not enough data, in situ data, that exist to validate such a thing. So we made use of a model, a very sophisticated one of the most sophisticated one actually, that exists, that is the Eco2 Darwin model. It's a global physical and ecological model that represents the world circulation is healthy resolving, so a very good resolution for all the physical oceanographic processes. It represents also all the biogeochemical layers, including primary production, and it represents not only the phytoplankton functional groups that we can detect by satellites, but actually it can give us the information of the whole phytoplankton community, because it simulates a bit less than 100 virtual types. And this is a dynamic model because it allows the ecosystem to evolve by himself and is constrained by real physiological parameters in the organisms, but also in the content of the nutrients and all the biogeochemical variables. What happened is that, how we can see here, I can show with the pointer. So in the first map here, we can see how from the model we calculated the same, but we could see if we had in situ sampling everywhere, so the whole local community, and these are the hotspots of biodiversity. If we calculate the same we could see by satellite in the model, we have this picture here, and here we can see where are the hotspots of biodiversity. So the relationship is going on, and we can actually identify where the hotspots are that are in this region. If we continue, so we saw that the approach was valid, it was possible. We have the application of the FISAL algorithm. It's not the only one existing, but is most probably one of the best. And we have the calculation of the hotspot of biodiversity at the global level using this approach. I was able to validate it on the only program that exists that runs since more than a decade, every year for 2 times a year from England to Patagonia, that is led by the Plymouth Marine Laboratory. And it gives information about the biodiversity in the water samples from phytoplankton, zooplankton, and also fishes. And I found a good relationship with the high and low patterns of diversity. From satellite, one advantage is also that we can have information about the whole temporal series. So we can calculate these maps, biodiversity hotspots, that change in spring, summer, fall, and winter, because of course in the temperate region we have the influence of seasonal cycles. And we can also calculate the persistent hotspots, so the regions where you always have during the year a persistent higher biodiversity. But I wanted to do more, because with biodiversity of phytoplankton, yes, it's very important for all the biogeochemical cycles and for the base of the trophic chain, but what about the higher level of the trophic chain that are mostly important for conservation and for commercial targets, for example? I made use of one of the most comprehensive databases about occurrences of different species, from fishes, marine mammals, invertebrates, etc. And on all these occurrences data, we calculate, me and my colleagues, species distribution models to have layers of the distribution of every species at the global level. And we distinguished between the trophic yields, so mainly herbivores, omnivores, carnivores, and top predators, to see if these biodiversity hotspots that we saw with satellites and that represent likely plankton biodiversity hotspot were also coincident with the biodiversity hotspot of higher levels of the trophic chain. And in fact, what happened was that if we consider, for example, the whole trophic levels, so invertebrate, mammals, fishes, from herbivores to predators, we have this picture, and we have this relationship where we can still identify these biodiversity hotspots. This is what we had before with the satellite view. And if we just consider the top predators, for example, this division becomes even more visible because these are the hotspots for top predators diversity identified using the satellite that we remember at the base is just an indication of phytoplankton diversity or environmental heterogeneity. An example of how we can use this kind of approach to delineate some areas for particular management or conservation is an overlapping of the highest possible values for the satellite index and for the other maps that comes from the species distribution models. And for example, if we choose 12% area coverage, it was the target for the Aichi a few years ago, we have 60% overlap with phytoplankton from satellite diversity and diversity of all the other levels of the trophic chain. So it means that, for example, if we choose these areas, we are able pretty much to conserve or to have the highest biodiversity of planktonic species and also more than 60% of the highest diversity of all the other species. This is something that was done also using other parameters by the conventional biological diversity that try to estimate biological or ecological significant marine areas. I highlight on total different parameters. And we can recognize that some areas are still present. Of course, there are different parameters and different area cover. There are projects going on now with whom I'm working at the Air Center with the MBON network. They are developing other maps like these ones with different approaches. And also, in particular, we submitted a NASA proposal that is aiming at pushing forward this concept of works. And they will use as background also all the work that I showed you. And we will use the most updated form of the PhySat algorithm, so more than 100 phytoplankton functional groups. And we will look more in detail at the trophic interactions. So, concerning the environment, I can say that the support of Earth observation technology is very important, and we can't neglect it at the point where we are, because satellites can give us synoptic and year-round information with a very good spatial and temporal scales, in particular for the pelagic environment. We can have also derived information about trophic interactions. We can have information about environmental shifts, climate change, effects of anthropogenic impacts, and also we can use it very well as a tool for conservation and management objectives. I thank you all. I didn't mention the part of enforcement, but I think this is also a very important topic on which satellite technology can give a very good support. So I'm happy to answer your question in the discussion afterward, maybe in English because I don't speak Portuguese yet. Thank you.